Tissue culture. It is the production or propagation of new plants from isolated plant cells or small pieces of plant tissue in a synthetic medium of culture solution. Plants are grown by removing tissues or separating cells from the growing tip of the plant and put in an artificial medium. The plant tissue divides to form small group of cells called callus. The callus is transferred to another medium containing hormones for growth and differentiation that forms plantlets. The plantlets produced are transplanted into pots or soil where they can grow to form mature plants. It is also known as micropropagation in vitro because it takes place outside the parent plant body in a test tube using an artificial environment. It is being used for the production of ornamental plants like orchids, dahlia, and carnation.